Everyone. Today we're happy to have Dr. Jia-Ren or Jerry Lin from Harvard Medical School and also from Laboratory of Systems Pharmacology, directed by Peter Sorger. And Jerry received his degrees in 2000 in Taiwan, in TU. Later, he did his PhD at Stanford in 2012. And after a year of postdoc at Stanford, he moved to Harvard to do laboratory systems pharmacology in 2013. And since then, he's been developing this technology called cyclic immunofluorescence, or for short, CyCIF. And he has developed the one for cell cultures, then later the one for FFPE tissues. And currently, he's applying these imaging and computation analysis to understand drug-gene interactions in cancers and using the high throughput approaches. And without further ado, I'd like to turn to Jerry.
Let me see. Thank you so much. Can everyone hear me okay? Just make sure. Okay. So yeah, thank you. So first, thank you for this great opportunity for me to present our work here. Yeah, it's sorry, I should join this seminar way years ago to learn all this interesting or know all the people here, but yeah, probably not too late by now. So yeah, today, yeah, the title here is a bit vague, but I think yeah, like I must just say, we try to introduce you a little bit about our technology and what's the work we've been doing with this we call the cyclic immunofluorescence. But since this is a spatial omics seminar, so we want to make it a little bit general. And just by looking at here, I don't know, maybe someone already familiar, this is a human tonsil tissue. And what you try to see here, it's only a 6 marker image, while blue is all the nucleus, so you can see cell here, but all the other marker maybe somehow label one of the cell type. And for example, CD8 will be the T cell and CD68 will be macrophage. And we have the Treg marker, FOXP3, and also checkpoint protein marker, PD1 and PD-L1. So by looking at here, actually, if you have the ability to probe multiple things at once, so especially with the special context. So that one thing you can start to learn from this is first, that of course, is the composition. How many of each cell you can try to learn from here. And second is what kind of distribution you can see here. For example, you can see most of the PD-1 positive cell were in this kind of region, the germinal center, as well as the PD-L1 is on the side, more on the epithelial cell. But also, if you go into more detail, you can start to see some sort of like cell-to-cell interaction. So I think that's why we have kind of emerged technology. So I try to list here. Sorry, it's not very up to date and it's not super extensive, but at least we try to cover some of we call the multiple image, especially is done by antibodies. So although yeah, we can call this spatial omic, but maybe a bit exaggerate things. We can probe multiple marker, but not to the level like several thousand like spatial transcriptomics can do. But in the other way, we can also have a more focused study using this multiplex, antibody-based multiplex image method. So like this here, although yeah, a great category based on what this can be achieved. For example, some of them is done by the fluorescence microscope, like our own methods, like immunofluorescence. Some of them will be require more complicated instrumentation, for example, using the mass spectrometry. Some of them actually using a quite different type of technology to achieve the same method to do the multiplex image. So then the great question or a big question is there a best multiplex antibody labeling method? And just my personal opinion here is probably not. And I put together here several things for consideration of how to pick a multiplex system, right? For example, multiplexing means how many marker you want to achieve. You want to do 10 marker, 50 marker, or even higher. That's in this case, at least some of the reference system here, for example, the narrow string DSP system that they kind of can achieve very high multiplexing. But what you lose here probably a little bit, at least right here, is the resolution, because you need to do almost like a region of interest, but not necessarily a single cell or subcellular resolution, as well as some of the system is limited by the sensitivity. So what kind of detection limit you can have, as well as the other things, although it's probably not so important for the research side, but kind of important for the clinical side, so how much throughput you can do. So you can handle a lot of slide or a lot of sample in one day, or you just do it very slowly. Of course, I know, yeah, everyone might be concerned is what kind of the cost for these things. Some systems much cheaper, and as far as we know here, I think the some of the method we develop here, the CySIF, probably among the cheapest method right here. And the only other method is somehow developed by commercial vendor. They do require a little bit proprietary components, so they will add up the price. Okay, so just, yeah, that's kind of quickly overview of the, we call the multiplex image field, and hopefully it's not too unfamiliar with the cloud here, since this is, I saw some of the work from Codex is present here, so probably a little bit familiar with that. But I just say back to our own research. So today I just try to, hopefully we have enough time to go over some of the points right here. So like how we develop this, we call the tissue-based CySIF or CySIF in general. There are something to do with the instrumentation, method, software, as well as the data analysis. That's all very important issue we try to bring up here. And also some of the ongoing project, either published or unpublished. And we try to introduce some of the work we're doing here. Hopefully can be the next generation multiplex method right here. And as well as a big project we are trying to achieve, and it's a joint project between multiple center, but like the title I list here, we try to build we call the tumor atlas, and what kind of the challenge we might encounter here. So yeah, so maybe go a little faster, but please stop me anytime if you have any question, or please just leave in the chat or try to email me later. I'm happy to take any question if we can. Okay, so first, I probably will scan this through since most of this is published data. You can find our reference. I actually have a slide list, all the key reference there. So we can share the slide on the web or through the video here, so you can actually learn that. But so cycloimmunofluorescence or CySIF, it's concept-wise is pretty, I would say pretty simple. Basically what you do is you just do a regular immunofluorescence just like you did before, but one key component here is you try to remove the signal after you've done the image, like the illustration we show here. And if you have ability to do that, and what we, the component or the solution we use is just fairly simple, is a very strong oxidation, hydrogen peroxide. So then you can remove the signal, but not necessarily remove the antibody. In that way, then you can relabel the antibody, but only the constraint here is you need to use the direct conjugate antibody to avoid the complication of the secondary or species issue. So then you can think about the same type of method can be used to the more high content manner to do a plate-based CySIF, or we even have a platform to do the, in a liquid sample, we can try to mount on the slide and to do the same way we call it immunoprofiling or liquid-based CySIF. And the good thing, or I think one of the advantages we use the similar principle with immunofluorescence is we can do either very low resolution like we demonstrate here. So it's a whole tumor scan. You can try to see the structure of the tumor, or we can close in to see the single cell resolution to see the fine structure. And even further, because it's just a regular immunofluorescence, so you can do a super resolution or super confocal or even a super resolution image using the same type of principle. So I think we haven't explored this further, but I think it would be really maybe useful, powerful cell biology tool if people start to using this more broadly. And just try to demonstrate our ability, although I would mention about some of the constraint here, but here is just a sample. We can try to cycle this 20 times. And each time, if you put in 3 different antibody, then we can achieve 60-plex image in one sample. So in some way, that's already, for our own research, is probably more than enough, since we're running all the available antibody we have in hand. But also the other combination or the other way you can use the decisive, since it's the immunofluorescence based. So you can combine with other method that's also fluorescence based. For example, here, you can do an RNA fish first on the left hand, you see here. Then on the right, you do a subsequent cyclic immunofluorescence, sorry about that. And you do a combination of the transcriptomic and the proteomic research. Okay, so that's basically some of the overview and the application we can use the cyclic immunofluorescence. And next, I just want to go into some more detail in terms of the image processing approach. Because in most of the case, we not just try to select a particular region of interest, but we try to get an overview or a very detailed view of a whole tumor. So normally we do a serious scan of the whole sample, like here is a melanoma sample in 20x, so we have about 100 frames. But if you think about the test, because we do a cycling process, if you cycle it 10 times, that means you have 100 image times 10 times. So in order to put all this together, it's not that easy a task in general, because basically not many people doing the sensing. So in a way, our software team actually come up with, or Jeremy here, he come up with a very clever solution. It's just by combination of the stitching and the registration together, then you can put together the image quite nicely to avoid some sort of the offset when you do the subsequent image. So in the end, we can do this kind of we actually demonstrate do an infrastructure on a web interface. So if you go to our website, I can show the link later. So we have the ability to do this kind of like hoof around, run in and run out, and switch different channels. So now you can have a different way to explore your data while you don't lose the, don't sacrifice either the resolution or the broad picture view of the data. And also go into a little more detail on the data analysis. Although, yeah, for this crowd, I think anyone might very familiar with how the image data analysis will be. But I just say they still quite challenge to us to try to convert the image data you see on the left to the digital data you see on the right. So the way we do it is using the segmentation that's mainly based on the nucleus signal. So what you see here is the green. So we can extract the region of interest of each individual cell. Then we can measure the signal on that region of interest. Here, I just demonstrate one of the marker. The green marker you see here is the tumor marker S100. And if you see the single cell heat map right here, that's kind of represent how you will get the signal from the left to the right. So once we get this data, it's not much difference from what you can get from the flow cytometry or other type of the multiplex method. Then the very powerful thing is you can try to put together some sort of marker combination. Like here is a cold biopsy of the breast tumor. So you can try to do a very interesting definition. For example, you can define the immune territory, tumor territory, and the other component, stroma component. So not only just try to do the things I first alluded, try to see the composition of the tumor. Now you can have this kind of the map to discuss what kind of the cell represent which domain, and also what kind of interaction there will be in together. But before we can go into more detail on the data. I just want to know, because all this method is antibody based, and it's very important to validate your antibody in the extent. That's one thing actually I found something quite surprisingly in the other antibody-based platform. It seems kind of hide behind the method. While we try to put the antibody validation up front, we actually have a paper that's only working on it, try to do the antibody validation. So here is just, well, how you can, so think about how to use this antibody. If, for example, for our research interest, we can do, we actually very interesting in tumor microenvironment. And as far as you can tell here, this is the hallmark of tumor. And for each individual big category, we can try to identify some important molecule. And if we have antibody, we can start to do this very thorough research on a particular question we are interested. And we divide our antibody, sorry, I should list it in front of my talk. But in our website, scisafe.org, we actually list some of the antibody we validate, and we test in a different level 1, level 2, level 0. But easier category is just level 0 is not working, and the level 1 will be somehow working, but in a very particular case, and the level 2 will be kind of general working antibody. And just try to say, we use a slightly different antibody validation approach, as you can tell from the antibody company, because and I forget to emphasize, all our antibody currently, although we use the fluorophore conjugate antibody, we try to get that directly from commercial vendor. We have some conjugation work in-house. It's been success, but the one thing we are thinking about is, if we are using the commercially available antibody, that means other people should be much easier to reproduce our work, rather than using some sort of more complicated in-house conjugation. Then they will, so we actually alleviate some of our antibody validation work to the company. But once we obtain a conjugate antibody from the company, we do several level of validation. Since we are doing in the tissue and the human tissue, some of the work we might not be able to do. For example, you cannot get a knockdown cell, but the basic characterization can be done since we can put multiple antibody in one sample. So we typically just try to see, using a very well validated antibody to validate the other less validated antibody. So for example, if you have assumption that, for example, the E-cadherin mymentin, they will label a specific location on your tissue. So you just try to observe if the antibody behave the same way you are looking at, as well as if you can go into more detail, subcellular localization. And here, sorry, maybe the resolution wise it's not so clear, but we use a nuclear lamine as an example. You expect if form a ring outside the nucleus, then you, by looking at that, you can know at least it should be expect to the prior understanding of this antibody. And for the last thing, and also maybe most importantly, since we are very focused on the immuno-oncology research, so we try to use our understanding with immunology. For example, here for example, if we thinking we have a good CD3 antibody, and the CD8 cell should be somehow all be CD3 positive in most of the case, then by looking at both correlation between this marker, we can somehow comfortingly assign that CD8 antibody we are using should be valid in this case. Okay, so but we actually go a little bit further by doing we call the validation through the clinical IHC. For some more important immune marker, we try to do is we run a clinical IHC, as you can see right here, using the immunohistochemistry. And then you can see the step standing. And we try to use the serial section while we're running the cyclic immunofluorescence. The red signal here represent the same antibody or same target you try to achieve in the IHC. Then we run through the multiple antibody, and we actually get a very good correlation between both site-safe code and IHC code. So that gives you another layer of the validation confidence, like you know what the antibody you are using is correct. So once you achieve that, then you can do a big combination of the approach. For example, here we just show in the myeloid lineage, we have come out with a panel that's labeled all the macrophage or myeloid lineage. And surprisingly, we found, although in previous research, people thinking some of them may be somehow overlap or redundant, but we do find a very interesting distribution. At least from the tumor case, we see somehow, although correlate, but not overly related or completely overlap distribution of this immune population. So that lead us toward our real research question. So here I just want to emphasize it's not just our work, it's our work with a lot of collaborator in the Boston area and actually across all the country. So yeah, thanks for everyone involved in here. But I just want to use, so first example here, yeah, most of this, sorry, yeah, this slide is a bit outdated, but this work has been published. So I probably need to refresh that. So again, I will quickly scan through so we have a little more time to talk about some unpublished data. But like here, we focus on a sarcoma that's with the TRK, NTRK mutation or fusion. Then we try to see how the drug response, so TRK inhibit, will affect the response in terms of the immune infiltration and the distribution. And just long story short, we see a very significant change before and after the drug treatment, and this somehow correlate with this mutation quite well. And also the other story is more on the melanoma side. And this is an extremely interesting case, a particular patient that goes through multiple different immunotherapy, and they get actually quite nice response, but they get the recurrent or relapse in a several different point. Then we've been very lucky to get the sample from multiple stage. So also in the same time, not just have image, but also the sequence data, DNA and RNA sequence. So we can cross map this two. And for the high dimensional analysis, we do see something very interesting toward, like we found a tumor specific marker in here is the NGFR expression. It's driven a bit like immune infiltration. So that could be a lead to an immune resistant mechanism on this type of patient. Okay, so yeah, I really want to go through some of the more detail in some unpublished data. So I will probably leave this two out, but the reference is here as well as we can share the slide later. So if you are interested, you can try to dig that up. So just try to quick summary what we think about our T-SISIF platform is. So like I mentioned earlier, so SISIF can be done in a high content, so culture cell. We can also do it in liquid biopsy. In here is the blood. And we also can do it in a FFP section. And I do not mention, we also done some work with frozen section, and it should be also doable. So then you can think about this. It could be a research tool that you can use all the way from the more preclinical stage to a semi-translational, and even in the future, hopefully we can use that in the clinical space. And you can think it can be as simple as like image cytometry using immunofluorescence, but also more importantly, it's the special context give you the power to dissect something that you probably cannot dissect with, for example, flow cytometry. So that's why the image is so important. Okay, so just try to quickly mention, it's still I hope it's a work in progress for everyone, since it's been like almost 5-6 years, maybe during the developer, and from just one person, myself, to a team effort. We just have our retreat before the shutdown. Unfortunately, we cannot have it this year, but now the group is grown to almost 50, and if you count the collaboration, maybe the 100 or even more. So yeah, it's kind of interesting field, and I'm really glad we have a chance to have this kind of collaboration. Okay, so just shift gear a little bit. Aside from the cyclic immunofluorescence, we are talking about the we thinking maybe useful next generation multiplex platform, and this is a collaboration with a company, small company in Seattle called RealSite, and they develop they call the Orion TissuePlex system. Okay, so go back to a bit background why we need. So as you can see, we try to advertise like CySIF has a lot of advantage, but I try to be fair. There are some disadvantage in the CySIF, or I would say for some of the cyclic method may suffer the same kind of issue is because you are cycling through, so it could be a bit slow. Slow means one slide process time could be slow, but maybe a bit counterintuitive, but not necessarily low throughput in a way that you can still scan a lot of slides in one day, but you just need to finish all the batch in a certain amount of time. So, but most importantly, I think our constraint here, since you do this kind of cyclic process, so you might experience some of the tissue degradation during the wash step or image step or all that step. And to say that also you have more step means more problem or more chance to get wrong. So it will be good to have some sort of, we call it one-shot system, just like you do for the image cytometry. You put all the antibody in one time, then try to resolve it by some sort of instrumentation. The one thing come in mind, or maybe some of you may familiar with the Akoya Polaris or their OPA system, and the principle is using the spectral omics or deconvolution. Although the deconvolution here, because you need to do a kind of the very continuous sampling and also a heavy lifting computational process. So I think in this system, so far as I know, they can achieve like a different spectrum or different type of spectrum or a marker to at most 10 marker, but not even further. So in RealSite, they come up with way to do the similar unmixing, although some of them proprietary, some of them in their patent, but they call the Spectra edge detection technology. So they can put 20 different fluorophosph or 21 in some case in one system and try to deconvolute that. And oh, sorry, yeah, I might be a bit gobbling here because I'm not the one invent the pattern, but principle behind is similar to the spectrum mixing you see earlier, but try to do a very careful selection of the spectrum as well as your excitation and emission. And by doing that, you can try to deconvolute that the signal is probably not supposedly be resolved in previously. So here is just show you an example. So in the system, we actually try to image 66, do an overlap image with 36 different channel. And as far as you can see here, some of them look really similar because they actually cannot, this is before you do the spectrum unmixing. So you cannot see too much difference between the adjacent channel. But after you run through the spectrum unmixing or the spectral edge detection method, you start to see the difference right here. So for example, I probably doing this wrong, but this should be a nucleus marker as well. Some of them is more the epithelial marker. Now you start to see the difference here. So our role here in the developer this system, not just troubleshooting, we actually help the company to develop the panel right here to specific research. So I can show you a little bit later what kind of panel we are working on, but it's again, the immuno-oncology panel we are working on. And one advantage of this system is also because if you are thinking some problem in the tissue image or the immunofluorescence image is the autofluorescence from the tissue could be problematic since they are somehow occupy the same same spectrum range. But because the way the spectral edge detection work, you can actually try to dissect out the signal. For example, this is a raw image without unmixing. And now if you can try to run through the same type of workflow, you can start to see the CK, the cytokeratin signal as well as the nucleus signal. But you tease out some of the background signal that show up here in red. So the fluorescence can be teased out by this case. And also in some cases will be interesting to learn what kind of the fluorescence signature here will represent. Okay, so again, we're using the same because the company work closely with us. We use the same type of web interface to show up the things we been achieved right here. So if you are interested, you can go to the company website to see what we been done. Although here is just a proof of principle, so it's just a tonsil case, but still it's a multiple marker combination. So now you can see what kind of the resolution you can achieve. And also a little bit toward the analysis side, the same type of analysis we use for SciSafe actually pretty straightforward. We can translate to this spectrum edge detection methodology. So we can now study a different, for example, cells to cell composition, or here I just using a very easy example, cell count. So then just very briefly, like in a germinal center, you should expect to see more of the B cell or proliferative cell, while in the outside of the cell, you should see some other marker. So it somehow work as we expected. So hopefully very soon we can push this toward publication, as well as the people can use this system to their own research. Okay. So yeah, sorry, yeah, this is the last part of my talk. So I think, yeah, hopefully we can still save some of time for discussion, but I just try to cover this a little bit, but not necessarily talking about whole the project, because this is, like I alluded earlier, this is a joint project and we only talking about a very small part of this project. And the project here, we call it TMB Sadhana Project. The original goal of this Sadhana project we try to do is we try to, since we have so many different technology, either spatial transcriptomics, spectral proteomics, or all the other methods, and also across several different institutes, we try to see if we can come up with a way to evaluate what kind of variability you will see among this different system, as well as even the different instrument or different institute when they operate these things. And also we try to learn a little bit more about what kind of error we will expect to see in this kind of the technology. So the setup will be like here. So basically we try to get a one sample, get a serial section, and then after that we distribute to different center. And as you can see here, we have about 8 different centers involved, as well as the 10 or 11 different technology being used. And today I will just focus on the things we've been done here at HNS. So we've been doing, we have a H&E slide as well as the SciSafe slide. So in adjacent, and also we come across the serial section that have 25 different sections from the same sample, and we just try to apply the same panel, and also it's adjacent H&E slide. So now we can use this. So the one things I did not mention is this being distributed within the H10 community, but hopefully we will release this soon for the broad community. So people can also use this as a reference set to try to evaluate their, hopefully is their analysis or their image processing procedure. So that would be a good reference set. But for us, once we get this sample, the one thing really draw our attention is just try to use one pair of the sample. So on your right is H&E clinical H&E stent. On your left is the SciSafe. Sorry, maybe I forget to mention. This is a colorectal cancer sample, CRC sample. So and with a little bit, if some people familiar with this field, we have some of the normal colon tissue left right here, and the rest of this big blue chunk region you see here is the tumor region. as well as we have nice stroma region, like here, the mucosa region. So then the one thing, even just by looking at 4 marker, including DNA right here, so you can start to see some of the marker actually distribute quite not homogeneously, while some of the marker is very extreme to some certain foci. For example, here the CD20 positive cell is probably only in some sort of germinal or tertiary lymphoid organ like tissue. So, and here is just another way to demonstrate the problem or issue we try to allude here is, this is just, we taking a 500 times 500 micron. It's probably what you get from the 20x objective image from the same sample. You can see how much difference you can get by looking at very field of view. So that's actually lead to a question or a challenge we try to discuss or try to emphasize in this research is, we try to do some sort of spatial analysis, but in a way we also do a spatial sampling in a way. So for example, if you try to do a field of view analysis, just most of time of view, then you basically just sample a very small region of your whole sample. And in a case that the overall distribution or overall composition may not be changed, but your sampling could be quite off from this overall or the real, I would say, the ground truth in this case. And it's not surprisingly, it's not just limited by the image itself, right? Because if you are thinking in most of the case, for the clinical specimen, we are trying to sample even smaller. A lot of people might familiar, this is how the whole section or FFP block should look like. But in some case, we do a congenital biopsy, so that's already a subsample from the whole sample of whole tumor you have. Or you can do that post fixationally, do a tissue microarray. So that's another way to do this kind of subsample. And we've been running the same kind of exercise since we have this opportunity to collect the multiple slide and the multiple section. So then, sorry, I might skip this one. We generate an idea we call the virtual TNA. So basically, you just do the same way people will do in the sample level, we can do it in the image level. So then we can generate this serious kind of the subsampling or spatial sampling. And then the question will be, how much this spatial sampling will reflect to the whole sample, right? So this is just a quick and dirty way to demonstrate. But if you're looking at this plot right here, so each the small green dots, oh, sorry, not green, small blue dot here will reflect, you can think about it's a virtual TNA or it's a field of view or whatever you are thinking about. That's how you can get from one sample for a particular distribution. In here is the, for example, CD31 positive cell supposed to be the endothelial cell. And then you can get, let's just take this as this example. You can get as high as like 70%, as low as close to 0%. But even you try to aggregate this, so let's say we aggregate all this, although maybe this is a bit exaggerated, we have taking 40 field of view, we get a mean right here. The true mean of the whole sample is show up here in the red dots. So it's still not quite that matched to the sample mean. And the other way to do this is once we get all data, we just subsample a single cell without any spatial bias. Even just by sampling several thousand cells, we already get very close to the true mean. That's the red dot right here. So as you can see here, the red dot and the green dot is matched much well, while the average of this field of view or TNA type of analysis will be a bit off. And this actually CD31 is the best case scenario. And if we taking something, for example, the CD8 positive T cell, so you can see here, you start to see some sort of systematic bias as well as a bit off, or not necessary for each individual. Sorry, yeah. In here, it's the since we have one sample with 25 different sections. So we just treat that independently. So increase our sampling power. But in fact, you can see here, you get a lot more bias if you're looking at a different marker. So if even you try to aggregate 40 different point of view, you only get a very correlation at most like 0.8. So why is that? Actually, it's quite easy or intuitive because all the marker or all the cell is not distributed randomly, right? Here we just pick 3 examples as a case. So for example, keratin in here is labeled tumor cell, and they tend to have their own territory, as well as the smooth muscle actin that's labeled a fibroblast or the some other muscle. The FOXP3 will be a Treg cell. So they all have, we call here the different scale of their spatial correlation. And via that, you can actually already try to calculate if you randomly sample or spatial sample, what kind of the variation you can get, right? So because it's done by just a very simple statistic principle, you can actually calculate how much we call it effective size of the sample rather than the total size. So if you do this kind of spatial sampling. So by using that theory, we can actually try to see how much random sample. So in here, random sample means you just get all the section, extract all the single cell data, and the sampling without any special information. While the other way to sample is doing a TNA type of sampling. And you can see that how much wider distribution you will get compared to the true like mean or ground truth here that's represented in the red line right here. So again, maybe since all this is virtual, so somebody might say that this is just not really, it's just due to some of our weird calculation. So we actually run through a real CRC or real colorectal cancer DNA. So this is done in collaboration with the Brigham and Women Hospital. So here we have 93 patients, and each patient will replace here as the 4 small core right here. And we pick from here, we only pick 17 whole section, that's or the block that we use for composite this TNA, and we will try to image again by image whole the section. So here is just a representative data, try to show you. So if we look at the whole section in a digital way, that's if we look at the CD8 positive cell, as you can see here, after we do a quantification, we get 8.3%. But if you look at each individual call, you can get as low as 4%, and some call can high as 43%. So it's a huge difference. Even you try to calculate the mean or try to exclude an outlier, there are no way you can get 8.3 here. And if you do a more systematic way to address this problem, this is just a quick figure you try to show here. So as you can see here, the red dot is the way how the TNA distribute, and the whole slide distribution is in here in blue. So as you can see here, not only you get more tighter distribution, but also you can see that their mean value also not always the same. Okay, and again, we can actually try to explain all that by this special correlation function I alluded earlier. So then what's the implication? So if it's just off a little bit, what's the matter? So like I show an example here. So if we just take the whole section data and try to evaluate, we use the one marker here, SNA. So try to see how much fibroblasts in this tissue. We actually can segregate out the survival curve quite well by using the whole slide. But if you try to get the same result using just TNA, you actually, although they show similar trend, but you don't have enough statistical power here to discern this. So in a way, and we do have a sample that counter the case. So in TNA, you see a difference, but you don't see that difference in the whole slide data. So again, we just emphasize that somehow you need to be careful that spatial sampling here could be dangerous, and you need to take your interpretation very carefully. Okay, so yeah, I think we are almost close to the end. So just try to recap what we mentioned here today. Like I say, we try to develop what we call a tissue decisive, and some of our project right here, as well as the Orion system we mentioned, that's the one-shot 21-plex system, as well as the project we involved lately, as most importantly is what we learned from this project. And just try to mention a lot of teamwork here. So this is a decisive research team here. Some of them I mentioned here, and some of them actually in the talk right here, Yuan Chen here. And also it's a collaboration from multiple labs. So here just show some of the PI here. And this is my PI, Peter Soger. And just the final acknowledgement, it's a lot of work from a lot of people and the funding resource right here. So, and again, thank you everyone for coming. And like I promised, I put some reference in the slide. So if you are interested, here is the reference. You can try to see if you are interested. And some of the protocol actually list right here, and some of the question we list up, like the human tumor atlas. And thank you for your extension. So I hope I still on time.
Thank you, Jerry.
Yes, I think we have 8 minutes left.
For this, yeah, fantastic talk. Any questions from the audience?
I think while the audience probably still thinking what question to ask, we have an announcement to make. So starting next week, I think we have to move to a different room link because I think this Zoom link is sort of like my personal Zoom conference link is no longer allowed for hosting this type of kind of public seminar. So here I'm gonna put the registration link. Okay, oh, okay, somehow I can. So I'm gonna find out the registration link and put in the chat box in the next couple of minutes. You can kind of click the link and register, and then you're gonna receive an email, which gives you the Zoom link you can use to join this seminar series. I think every week in the same link you can use for yourself. Okay, everyone has different link. Don't share with each other. All right, so maybe back to the Q&A session. So Ahmet.
Yeah, any questions from the audience? Feel free to unmute or type on the chat. So one question while others are thinking is, Jerry, so the cyclic method, right? How robust is this between the cycles? Like how much of the, I mean, I know that in your papers you quantified, but do you have to do this optimization for every experiment, or this is generally very successful if you use those bleach like for 1 hour, right? And things like that, is it gonna work or not?
Yeah, that's actually a great question. And that's the review comment we got when we first published the paper. So I would say, like you said, we demonstrate in the paper that in most of the case, a lot of antibody did not affect by cycling. So we can get a pretty robust result. But the other way we attack or thinking about this question is, if you try to do a comparison among the sample, if they running through the same procedure, even you thinking about there are some sort of like almost like a degradation or deterioration between cycle, but since you are compare, hopefully the same type of degradation will apply to the same the batch of sample you try to compare. So in that way, you don't suffer too much from that. But yeah, it do raise a question that some of the marker we think might have more problem if we put in a later cycle. But to this, sorry, to this question also, the other things is I think maybe I did not say it clearly enough. The tissue degradation is another or even more important issue. If you lose the tissue, for example, in a cycle 9, then the later cycle, this sample won't work, right? So that's why I think that's why we are thinking to introduce or develop the second or complement approach that I show here, the Orion approach. that will help because it only do one shot, one image cycle, and it's all done. And also we did not say, I did not say it explicit here, but you can actually cycling after that. So once you done a 20-plex image, you can continue with the decisive workflow to do a rest of the cycle. So that's give you opportunity to add on top of the 20 press and do multiple marker. So I think that's there are some opportunity there we can explore even further.
Thank you.
Yes.
There's also another question. Do you think there is also sampling variation when going from 2D tumor cross sections
Yes.
to 3D?
That's a great question. Yes, that's we did not demonstrate that clearly, but I want to see if I think everyone still can see my slide, right? There are some, yeah, there are, we indeed expect there are some like a variation from 2-dimension to 3-dimension, as you can see here. So sorry, yeah, maybe it's a short amount of time, so try to gain too much information, but this is a serious section across, almost like across Z. And each section apart is probably 25 to 30 micron. So from the first section to the end of section, you actually try to survey about 500 micron difference. So that do show up some difference for some marker, like here you can see the endothelial population to change a little bit. If you just follow, okay, sorry, you need to follow just the red dot because red dot is the mean of the whole slide. But in any case, if you try to think this way, in any case, this variation won't be bigger than you do a sampling like a very small range. For example, if you look at the distribution of all these red dots versus the distribution of, we just pick any one of them, like any small blue dot right here, you can see a lot of variation. So again, it's not surprisingly because if you are thinking for the whole 2D space, you are across several thousand micron, because that's the millimeter range of the tumor. So from the Z section, you do tend to have less of the distance. So you expect less, but I think that's an interesting question, and we will learn even more when we have this kind of data set that we do have the way to sampling more. And here is even better example that if you look at some cell distribution, for example, T cell, it seems quite stable over the slide. You can see here. So, but it do still suffer from the sampling if you do a very small region of interest. Yes. Thank you. Yes, thank you for the great question.
Any other, maybe final question? Jerry, I think has one more minute.
Yes.
Yeah, I think, Jia-Ren, I know you have to run. I kind of want to follow the question Ahmet asked before, this kind of technical question. So people actually in the single RNA sequencing field, it's a kind of very thought-provoking question. So they ask me, so can you sequence in the same cell twice and you see how reproducible your data is? So now I realize actually you can do, but now sequencing, you can image the same sample twice using exactly the same antibody, and then you get the answer, right? So
yeah, we do that. That's what we did in the paper. Although in this case, since you did not remove the previous run of antibody, so then you have some competition going on. So, but for the antibody we've been tested, if we pick a really abundant protein, I think in the paper we use something like vimentin, and you barely see a difference if you repeat it at this antibody. So that's one comforting way. But indeed, we need to answer this question even better with some other technique. Yeah, but I think so far in the market or in the field, there are more and more this kind of technique. So it will be really interesting. We have a counter or the across platform comparison and address this question even in a broad scope.
Great. Okay. Thank you, Jerry.
Yeah, sorry, have to run, but yes, thank you everyone. And like I say, I can try to distribute the slide later to you. And if you have any question, I'm welcome to answer through email or other means.
Perfect. Thank you again. And next week, guys, we have another speaker from Institute for Cell Science, Dr. Vanhani Gunawardane. and she's going to talk about stem cells and RNA fish. And we look forward to seeing you guys. And have a nice weekend. And
yeah, thank you. Also, yeah, please be aware we're going to use a conference pre-registration system. Make sure you register before you come over here. Thank you, and see you next week.
Bye-bye.